Hello, everyone, and welcome to today's lecture. And so today we're going to be covering a few different topics, all related to the genome, including horizontal gene transfer, gene regulation, and genome evolution. Just as a reminder, your first exam is coming up. It will be on October 7th. So this is an exam I will send out. It'll be about 40 multiple choice questions. You will have a few days to do it. It'll be sent via email, and it will be in a Google form for you to fill out. And you'll get your results exactly when you do it. So once you submit that final question, you are all set. And you'll know your grade right away. Now, in terms of this exam, you get one shot at doing it. It is open book. If you're going to work with someone else, I can't really do anything to stop you from working with someone else, but it is definitely open book, open resources, so you only get one shot at it. Now, it's going to be a difficult exam because you have all the resources of the lectures and the internet at your disposal. So it's not going to be just a cakewalk exam because it's fully remote. So just as you know. Now, the final thing that I'd like to mention before we get started is you need to decide your topic. So most of you formed your groups. That's great. You do need to tell me your topic. You can do this either via email or you can do it in the sign up sheet that I sent around. You can just dump your answer in there and and I will sign off in that sheet. I've already signed off on I think about 8 or 9 of your projects already. And so for those of you that have already let me know, you know, you're all set. If you do need help deciding on a topic, let me know. Be happy to chat about it. And the other thing I'd like to mention is when you're doing this, make sure you take a much more specific topic. So don't try to take a whole genera of bacteria or an entire group of organisms, say like, protists, right? That's just too big for a microbe wiki. You want something that's more specific. About the measles virus. So for those of you that were paying attention last year, you would have heard the measles outbreak here in Boston, and you probably heard about it in Washington State, and you probably heard about it in New York State as well. And you can Google measles outbreaks, and you get all sorts of results for this. But we did have a measles outbreak here in Boston last year, and that's kind of a scary thing. And it's scary not because just because measles is scary. It's scary because measles, as we'll talk about, is essentially an eradicated disease here in the United States. So let's talk about measles. Now, measles is caused by the measles morbillivirus. It was formerly called the measles virus, or MEV. It is a type 5 virus, and we'll talk about what that means later in the class when we talk about viruses. We're going to talk about a very specific way to classify viruses, which is called the Baltimore classifications. But more specifically, measles is an enveloped single-stranded RNA genome. It is negative sense, and it is a round virus. Just as a note, this is actually pretty similar to COVID-19 as a virus. And so we're going to talk about COVID-19 later in the semester when we talk about epidemiology and public health. But do know they do share some similarities in terms of their structure, not in terms of their evolutionary history, but in terms of their structure. And it is a member of the viral family Paramyxoviridae, which also includes the mumps virus, which is an equally is not as fun virus. Now, measles has a very storied history with mankind. Now, there are best estimates at this point put the evolution of measles from the rinderpest virus around 1100 to 1200 AD. Now, rinderpest is a cattle plague. So what this tells us is that very much like COVID-19, measles is zoonotic in origin, meaning it jumps from cow to human, very much like COVID-19 jumped from bat to human. And so this is approximate dates, about 1100 to 1200 AD. It might be around longer. It might have been around shorter, but our best estimates at this point, again, is about 11 to 1200 AD. Now, measles has this history in modern times as being a relatively mild virus, but it is an absolutely brutal virus when it's introduced to a brand new region. And just to illustrate what I mean by this, in the 1530s, it actually killed half the population of Cuba and Honduras, and then eventually swept across Mexico and Central America when it first got there. And then it killed, when it reached Hawaii in the 1850s, it killed 20% of the Hawaiian population. So this is not a mild virus as it's made out to be. This is a extremely, extremely dangerous virus. And it's dangerous when we haven't encountered it in a while. And that's the case in what we're seeing in Central, South America, and the Caribbean. And it's also the same thing we saw in Hawaii. Just hadn't encountered it, or it's been a while since it had been encountered. So it's dangerous. Don't let anyone tell you anything different about measles. It is a very, very deadly virus. Now, the cool thing about measles, and it's just a Boston thing. So if you're not from Boston, this maybe is less cool for you. But it was first isolated at Boston Children's Hospital in 1954 by Dr. Thomas Peebles, and he obtained his virus from a 13-year-old student whose name was David Edmondson. Eventually, all the virus that Dr. Peebles collected would be isolated and used to create a series of vaccines, which we now have improved on, but was one of the sort of the most important vaccines that we developed through time. And we're going to talk about vaccines later in the semester, but just to know, the measles vaccine was a huge um, deal when it happened. Now, how does measles transmit? Well, it's a virus that's found only in humans, which means you can't pick it up from your cat or your dog or a cow or a bat. So there's no animal. Sorry, there's no known animal reservoirs. Which, if you're trying to eradicate disease, it's actually pretty good. One of the reasons it's really hard to get rid of uh, Ebola in Africa is because Ebola has a reservoir in bats, and it's hard. You can't really kill all the bats in say the Congo basin because bats are super important. So diseases that have animal reservoirs are typically harder to eradicate than those that do not have animal reservoirs. But measles, again, does not have an animal reservoir, which is good. The transmission is airborne or via close contact, and it's typically taken by coughing and sneezing. And measles is kind of a tricky virus. It can survive on air and surfaces for hours. Um, and there is some evidence that suggests that it might be able to live for days on surfaces. So kind of a pesky virus, but definitely for several hours after transmission. Once those people are infected, they can spread measles from four days before they exhibit any symptoms, all the way to 4 days after a rash appears. And we'll talk about the virus symptoms in just a moment. But measles is a very, 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 very contagious disease. If we were together as a class, there is about 20 of us in this class. If I had measles, because of how contagious measles is, just us being in the same room and me talking with you guys, all but basically two of you would catch measles from me. That's how contagious measles is. 90% of susceptible people will get it. So it's really, really contagious. Um, and if you were paying attention when this happened in Boston, you would have seen news articles published by all the class that transmits. Now, in terms of the pathogenesis of measles, it targets your epithelial cells, typically in the trachea or the bronchi. So you're thinking of your throat. It binds via a protein called hemagglutinin, which is, you can look at this diagram up here of the evil measles virus. It has the H protein or hemagglutinin, which binds to your epithelial cells. And what it does, like all viruses, as we'll talk about later in the semester, it binds to specific receptors on your own cells. That's how all viruses work. And what ends up happening is the virus binds and then it ejects its genome and it reprograms your cells to make more viruses. And again, we'll talk more about viruses later in the semester, and this will become much more clear once we do that. But once it infects your epithelial cells and again in your throat region, it is then carried to your lymph nodes. And this is typically done by infected dendritic or macrophages, so parts of your immune system. And now, for those of you that have ever taken anything about learned anything about immunology, you'll know that the lymph nodes are really the garbage dump of your immune system. And once things make it to your lymph nodes, they can actually be carried anywhere your vascular system goes. And so once it makes there, it further spreads to more lung tissue and then spreads from lung tissue to other organs, your intestines, and even your brain. So measles, once it makes an infection, it's kind of like COVID in the sense that it hitches a ride on your blood to everywhere. But it does, its first stop is your lungs, then your lymph nodes. And once it hits your lymph nodes, it makes it to wherever it needs to go. Now, the symptoms of measles are pretty, they're not particularly crazy, but the first signs are typically a high fever. So you're pushing, 104 degrees or 40 degrees centigrade. This begins about 10 to 12 days after exposure, and it lasts between 4 to 7 days. Next up, you get some cold-like symptoms, a runny nose, a cough, red and watery eyes, and then you develop what are called Koplik spots, which are these white spots inside your cheeks. And after about 3 days on your of a face and upper neck rash, it spreads, eventually reaching your hands and feet. And then this rash ultimately lasts for about 5 to 6 days and then fades. On average, you have a rash about 2 weeks after the exposure of the virus, and it can range from 7 to 18 days. It just depends on the person. Um, what other complications are associated with this besides the symptoms? Well, there is diarrhea, which is never fun. Diarrhea is not such a big deal in more developed countries, but as we learned from the massive hurricane in Haiti, I'm sorry, earthquake in Haiti in the 2010s, diarrhea from complications is can be very, very deadly. So not a big deal here in the United States usually, but in less developed places, diarrhea is a big and bad symptom, and it's about 1 in 10. Next up is actually acute encephalitis. And for those of you who've never heard of encephalitis before, if you've always wondered what encephalitis is, it is simply just swelling of your brain. And when you swell your brain, it only has a limited amount of space to swell. And once you cause your brain to swell, you typically induce permanent brain damage. And this occurs in about 1 of every 1,000 measles cases. Now, remember, measles hops on your lymph nodes to the rest of your body, including your brain. So it hits a lot of your organ systems, not just your skin, as you're seeing with symptoms. Um, it's pretty common that those that are going to perish from measles typically die from a pneumonia or some sort of neurological complication. And this is about 1 to 3 out of every 1,000 infected children. So it's not a super high mortality rate, but it's certainly a mortality rate nonetheless. And then the final thing about measles, and it's probably the scariest thing about measles, and it's something we're actually learning a lot about, is this really rare condition called subacute sclerotizing panencephalitis, or SSPE. Now, SSPE is a degenerative disease of your central nervous system. And the idea is about 7 to 10 years after you're infected with measles, what ends up happening is the person develops some pretty severe intellectual deterioration, followed by seizures that eventually lead to a death. So you could picture this: pretend you're a 3-year-old, you get measles because you weren't vaccinated. When you are, say, 10 to 13 years old, you develop this thing and you abruptly die. So you're dying 10 years after the fact from measles. And I think this is probably the scariest part about measles, is that you could get measles, survive, and then go brain dead in 10 years. So it's a really sort of scary phenomenon. And there's also this other really scary phenomenon with measles is measles has been known to actually wipe out your immunity. So measles does this weird thing. It will reprogram your immune system. So pretend you get measles and you had, say, 10 flu viruses before you got measles. What measles does is it wipes out all immune memory of those 10 flu viruses you got before you got measles. So kind of a weird thing. Measles is just a weird bug, and you got to remember this is a virus that's been evolving with humans for about 3,000 years now. So it has had a lot of time to learn how to mess up the human physiology. So you might be asking yourself, how do you get diagnosed with measles? Well, in the United States, it's not too common, but it's becoming much, much more prevalent, as those news articles hinted. But it's typically diagnosed with consistent and clinical presentation of the complex spots and a rash. It is confirmed by a detection of a measles-specific IgM antibody in a serum, or the measles RNA by a technique called real-time polymerase chain reaction, or RT-PCR, in a respiratory sample. And so that's how it's. We'll talk about this RT-PCR later in the semester, but you're just basically testing whether or not measles is present or absent in your body, right? You can't really have measles inside you without being infected with it. So if you have measles present, you got measles. And the thing about measles is there's no specific treatment for measles. Just remember, antibiotics never work against viruses. Antibiotics only target bacteria. So you can't take an antibiotics for the measles. You can't take an antibiotic for the flu. You can't take an antibiotic for COVID. You can only take an antibiotic for bacterial infections. And despite how prevalent measles is, there's no effective antiviral drugs that have been discovered, but there is a promising one actually currently in development for those people that do contract measles. But the prevention for measles, and the reason why there's not many meaningful medications for it, is because of the vaccination. Vaccination is super, super effective for measles. So the measles vaccine was developed by a gentleman named Maurice Hillman, and his vaccine came available commercially in 1963. 5 years later, he improved it. And it's a really great vaccine. It is super effective. You get it twice, and you're immune from measles for your entire life. And we'll talk about how vaccines work later in the semester, but just know this is a really successful vaccination. And this vaccine alone is estimated that it saves about 1 million people's lives per year. So, you know, kind of a big number. A million people is a lot of people. But that being said, there are still cases globally. As I mentioned, there's some here in the United States. But when we look at them globally, we have a map of the world. Dark red colors to greater than 1,000 incidences of measles. White is basically no measles, and gray is no data available or reported. And what you'll notice is that the vast majority of the world is pretty light colored, right? Most of it's not that dark red. But there are some hotspots: the United States, Brazil, Africa, large parts of Asia, right? So measles still exists as a whole. So this is data from 2019 over a 6-month period from the World Health Organization. So measles is in low prevalence right now because of the vaccine, but it's still, it is still present across the globe. Now, in terms of measles outbreak, measles as an endemic disease was actually eradicated from the United States in 2000. But the way we get measles in the United States, at least historically, was reintroduction from places that have measles natively, so like Africa and parts of Asia. For instance, the 2018 outbreak in New York, for example, was actually caused by someone bringing it in from another part of the world to the United States. And so measles was eradicated from the United States at one point in time. Now, Massachusetts has a really, really high vaccination rate. We do a really good job here with medicine, as you guys probably all know. Our vaccination rate is 98%. So if measles got to Boston, it's not likely to spread because we have a very high vaccination. Of measles spreading. So we're basically not eradicated anymore. Now, we can look at annual measles cases in the United States from 1944 to 2019. We could see this is a million cases reported. That's the scale. This is a logarithmic scale. And then we have time on the x-axis. And you can see we have million cases per year, and there's some variability year to year. And you can see the first vaccine was approved, cases dropped dramatically by almost 2 full orders of magnitude. We introduced a new vaccine, and it drops dramatically again. Then it starts coming back up again. The CDC steps in, says, hey, you have to start vaccinating twice. And vaccination rates continue to climb while the infection rates drop. Now, there's an important milestone here. There's that myth that vaccines, in particular the measles vaccine, causes autism. That happened in 1997, and you can see after that we hit an all-time low in the early 2000s, and then things have started to gradually pick up again. So not a pretty picture in the latter part, but again, you can see how effective this vaccine was for just essentially eradicating measles. 37 cases is nothing when you are starting out at close to 1 million cases. It's a really, really powerful example of how vaccination works and how powerful vaccination can be to curbing the spread of a disease. So it's really, really great. But as I mentioned, this anti-vaccination movement is causing measles to rise again. We finished 2019 with about 1,375 cases reported here in the United States. Knowing about science is really opening that book back up, which is kind of a sad thing, but I digress. So let's move on. Let's move on to the topic of the day. Move on to something less depressing than measles coming back. And this is going to our first stop is going to be horizontal gene transfer. And now the first questions we're going to answer is, how do prokaryotes transfer genetic material? What types of genetic elements move within and between prokaryotes? And finally, how does DNA get transferred between domains? So when we're thinking about canonical gene transfer, we think about vertical gene transfer, where parents, whether it's a human or a bacteria, are sending their DNA to their offspring via reproduction. So in the case of bacteria, vertical transmission is a bacteria divides, replicates its genome, and passes its genome to both of its offspring. So that's vertical transmission, mother to daughter cells in the case of bacteria. Now, horizontal gene transfer is the movement of genes between cells independent of that cell cycle. And the idea being is that a cell can acquire DNA from the environment or its neighbors after it's done its cellular division. So no bacterial reproduction required. And so these are two types of gene transfer, but I'd like to just make note. We're going to talk about horizontal gene transfer. And what we know about horizontal gene transfer is mostly from studying bacteria. But there is increasing evidence actually that archaea have similar amounts of transfer. But just to know, what we're going to be talking about was learned from bacteria. Now, horizontal gene transfer has some pretty significant and immediate effects on human health. So we have just a very, very simple example here. We have a bacteria. This bacteria is Shigella. If this bacteria gets infected with a virus carrying a specific toxin, it can then become a different strain of bacteria, E. coli, I'm sorry, a different species of bacteria. And now this bacteria has a diarrhea-causing gene, which makes it more pathogenic, which makes it a more deadly bacteria. So we started with a harmless bacteria. It acquired fast and how powerful horizontal gene transfer can be in your everyday life. So in terms of horizontal gene transfer, we're going to be talking about three ones today. The first is transformation, which is the direct uptake of free DNA from the environment. Transduction, which is DNA transfer mediated by bacteriophages or viruses that infect bacteria and bacteria only. And then conjugation, which is direct cell-to-cell transfer of DNA. So let's talk about transformation. Now, transformation has a fun history. And very much like the very first lecture where we talked about the history of microscopes, I'd like to just tell you a little bit about this really cool discovery of horizontal gene transfer by Frederick Griffin. Now, Frederick Griffin wanted to make a vaccine for pneumonia caused by the bacteria Streptococcus pneumoniae. Now, you might recognize strep is a name that you may have heard of if you ever had strep throat. This genus of bacteria is pretty well known to cause diseases, including pneumonia. But strep has also been known to cause throat infections, such as strep throat. So if you ever wonder where strep throat got its name, it got its name from the jerk of a bacteria that causes it. Now, Strep pneumoniae has two distinct strains, same species, but two distinct strains. The first is the virulent strain. So when we say virulent, it means it's a bacteria that has the capacity to cause infection. Now, this virulent was smooth. It was an S strain, so it looks like this. It's pretty, globular, very, very smooth looking. It has and it's a capsule. So if you remember back from when we discuss bacterial morphology, capsules are sort of shiny protein coats that protect the bacteria from antibiotics and the immune system and all sorts of things like that. So it makes the bacteria a better pathogen. And if you infect a mouse with this virulent or S strain of strep pneumoniae, it kills mice. And there's a second strain of this, which we call the avirulent or the rough form or the R strain. Avirulent is obviously the opposite of virulent, meaning this one does not cause disease. And you can see what this R strain looks like relative to its S strain. And I hope it's pretty clear that you can see that this one has a capsule, this one does not. And if you infect this R strain into a mouse, it doesn't kill the mouse. So this is the backdrop by which Griffith did his work. Now, what he did in 1928, he took two strains of strep pneumoniae. What he did is he killed at the S strain, which is the bad strain, and he injected that into the mouse. Now, so normally, if you have the S strain, you infect it into a mouse, it kills the mouse. But if you take the S strain and you heat kill it and you inject it into the mouse then will not die. Next, what he did is he took the R strain and you infect that into a mouse, and the mouse doesn't die. Then what he did is he took the S strain, he heat killed it, looked it under the microscope, and then injected it into a mouse, and the mouse itself didn't die. But the cool thing is what happened is when he took the heat-killed pathogenic strain, combined it with the non-pathogenic R strain, again, this one is heat-killed, the S strain, injected into a mouse, he would find that the mouse was dead. And what he would recover out the back end was the S strain. Now, you might as just a person think, well, maybe he contaminated this, right? Maybe he accidentally gave his mouse the S strain instead of giving it the R strain or something like that, right? But he repeated this over and over and over again, and he kept getting the same results. So what was going on here? He was taking a bacteria, killing it, then it wasn't able to kill the mouse. But when he combined the deadly bacteria that was killed with the non-deadly bacteria, all he would recover at the back end was the deadly bacteria. So what the heck's going on here? Well, this is the process called transformation. And transformation is the uptake of naked DNA directly from the environment. And the thing about the environment is no matter where you are, it is chock full of DNA, whether it's on your skin, whether it's in your gut, whether it's in the soil, whether it's in the ocean. Things die. And when they die, they release their DNA. And there's also this weird phenomena where cells will actually release DNA on purpose when they're living. It's this weird like evolutionary thing that is weird, and I just don't want to talk about because it's weird. And the thing about it is, once that DNA gets out, whether it's by killing a bacteria or on purpose, it can actually remain intact for a very, very long time. And what do I mean by long time? I say thousands of years, and that's true. When we can find DNA that's thousands of years old, preserved in the environment, commonly. It's pretty amazing. DNA can survive, is very, very strong, it's very stable, it can last the environment a long time. And the environment is chock full of DNA. Now, naked DNA, which we typically consider what is uptaken by bacteria during transformation, is typically fragments of chromosomal DNA, or what we would call a whole plasmid. And we'll talk about plasmids a lot more later in this lecture, but ultimately to get it, a cell must be what we call competent. And all competence means in a bacteria is not they can read or anything like that, or they can do their job, right? It's not like how you describe a human as being competent, but competent for a bacteria is simply the capacity to uptake DNA from the environment. Now, transformation as a process is induced differently in different species of bacteria. So in our gram-positives, so those are the ones with the very thick cell walls, they secrete what's called the competence factor, or the CF. And as CF builds up and the population grows, it actually induces the expression of what we call translocosome proteins. And these translocosome proteins actually physically uptake DNA. So as a bacteria grows, it's actually stimulating itself to pick up DNA from the environment. In gram-negatives, so with the small cell walls, They either induce competence under stress, typically via starvation, but could be a couple other different ways. Um, it's either through stress or they're always competent, so they're um, always ready to take up foreign DNA. And then finally, we can also have what we call artificially competent cells, and these are things you can do in a lab, right? You can induce that by heat or chemicals or electricity, and these are important for molecular biology and molecular biology tools. Now, so let's talk about transformation. Now, we have natural transformation here and then artificial transformation here on the right. Natural transformation is we have naked DNA in the environment, and a bacteria encounters it. It has proteins to uptake it, so those are those translocasomes. It uptakes the DNA from the environment through the proteins, brings it inside of the cell, and then integrates it by homologous recombination. So this should ring a bell for how chromosomes cross over when you learned about meiosis in your very first science class here at Roxbury. But it incorporates via homology of the naked DNA to the genome of the new bacteria. So that's what happens naturally. Artificially, you can force pieces of DNA into the cell via, again, by heat or electricity or things like that. And then we rely on the integration by homology as well, or the plasmid itself to just do what it does. And as I mentioned, plasmids, remember, they're autonomous. They self-replicate themselves. So we talked about that last class. Now, let's talk about gram-positive bacteria first. They're much more involved, because one of the reasons they're partially involved is just because of how thick their cell wall is. Now, as I mentioned, as these gram-positive bacteria are growing and growing and growing, they build up these competence factors, and these competence factors are short 15 to 20 amino acid peptide sequences. Now, as CF accumulates at high density, it binds to what we call a kinase, which is COMD. And once COMD does its thing, it essentially phosphorylates COME, which is what we call the competence factor action, which is basically just a DNA binding transcription factor. So it regulates how DNA is expressed. And once this does, it activates a whole series of genes and operons to essentially trigger the direct uptake of DNA from the environment. But again, it all starts with a series of proteins here, COM-A, COM-E, and COM-C, and some energy to export and produce competence factors. But once there's enough, it then induces COMD to change the behavior of the bacteria. So you might be asking yourself, why do we care? Well, in the case we talked about with Griffith, right, we took a non-pathogenic strain. It was competent. It uptook DNA from the pathogenic strain, and then that bacteria itself became pathogenic. So it's a particularly useful thing if you're in a clinical setting for a bacteria, because if you could become a better pathogen, well, it means you'll do better, right? You'll reproduce more. You'll be more successful. But in terms of why import DNA from the environment, there's lots of good reasons. DNA is an important food source for a lot of bacteria. It's got lots of nitrogen and phosphorus, which all bacteria need. DNA is also useful for fixing your own genome. So say you're living in a population of similar bacteria and you have a damaged piece of your own genome. Well, it's instead of fixing your genome, it's easier to pick up DNA from the environment from a bacteria that's closely related to you and pick it up and incorporate it into your own genome to fix it. So it's an easy way to fix your genome without expending any energy or any resources. And then finally, it's actually a really important source of genetic diversity. It brings new genes into the recipient. And it might mean that you're bringing in genes from people that reside in the same environment that are more successful for you. So you might acquire a new trait, and that might allow you to maybe say, eat a new food source, or potentially produce a new antibiotic, or be resistant to a new antibiotic. So it can bring a lot of important things to a bacteria. So that's transformation, again, direct uptake from the environment. Now, next one is one that's actually something that happens to humans as well. This is a process called transduction. And this is a process mediated by viruses. And so we're talking about bacteria. So we're talking about bacteriophages here. And what ends up happening is transduction involves the accidental packaging of the host DNA into the viral capsid. And this is something that's estimated to occur about 1% of all new phages produce. And very much like our transformation, it is essentially injected into the chromosome of the host via recombination. So let's talk about how this works with this French figure. If you're wondering why it's bacteriophage and bacteriae, it's French. And when you see ADN, it's DNA, but that's just the way the French write it. Normally, I don't use figures from other in other languages, but I really like this figure. It's a really nicely designed figure of transduction. Now, we have a bacteria here. A bacteriophage now attaches to it. It injects its genome here in pink. And what it does, it causes the bacteria itself to start reproducing copies of the phage's genome. And that's how viruses work. They inject their genetic material and they have the host produce brand new viruses. Now, what ends up happening is, oops, I'm so sorry about that, is it produces pieces of not only the virus's genome, but you can also see there's pieces of the host genome. And so what ends up happening about 1% of the time, as when you produce new viruses, about a small portion of the host genome, so in the green, is incorporated into the virus's genome here in purple. Now, once the virus kills the host, it escapes the host into the environment, and then the virus goes and infects another bacteria. So you can see this virus, it's got its own genome, and then part of the bacteria's genome. So it ejects its genome, and what ends up happening is, remember, some viruses don't kill the host right away, right? They go into that lysogenic cycle. So what ends up happening is the virus injects its DNA plus the bacteria in the first part's DNA, and that DNA ultimately gets incorporated into this bacteria's genome. So this bacteria, via this virus, is acquiring a gene from a completely different bacteria. Now, just to give you an example of this in real life, and then I'll talk about an example of this that's pertinent to your everyday life. When it's not just being a normal marine bacteria, it can be pathogenic. And the way Vibrio cholera becomes pathogenic is by being infected by a phage called CTX-5. And this phage actually carries the gene for the cholera toxin. And so the toxin genes plus what we call a pathogenicity island allow for enhanced adhesion of Vibrio cholera to a human's epithelial cells in your gut. Transduction, one instance of transduction viruses. So this just doesn't happen in bacteria. This is actually something that happens to you as well. Just as a note. So that's transduction. Let's move on to the final type of horizontal gene transfer. And this is the much more directed version of horizontal gene transfer that we call conjugation. And this is through direct cell-to-cell physical contact. And as you can see here, we have bacteria here, and they're physically connected by this, what we call a pilus. So this is the essentially the version of bacterial sex. It's not like sex, sperm and egg, but it is direct physical contact to exchange genetic material. And this pilus itself actually exchanges the plasmids. So let's talk about this process as a whole. Now, conjugation typically involves the movement of plasmids, or what we call transposons, or pieces of DNA called transposons or transposable elements, but it never involves the transmission of whole chromosomes. It's typically just small pieces of DNA. Now, the cool thing about conjugation is it's typically what we call self-transmissible, meaning that plasmids typically carry all the genes needed for conjugation to occur. This includes the genes for the pilus, the mating bridge, and DNA export. And this is carried out by, um, and so I'm sorry, this is an example of this is what we call F plasmids, which contain the fertility factor or the F factor. So let's talk about this process a little bit more in depth. We're going to ignore this sort of gross stuff on the right on the right here, and we're going to focus on sort of the big process here. So we have a bacteria. It has a plasmid. It is an F positive cell, which means it has the F factor on its plasmid. The first thing it does, it extends its sex pilus from the left side here to the right side here. Now its recipient is an F minus cell. Now what happens is a relaxosome, which is just like a protein complex that essentially retracts this pilus. It just pulls the other bacteria nice and close. And then this relaxosome essentially causes a bridge to a more direct contact and eventually a much more direct bridge to form. Then what ends up happening is the plasmid itself, remember the plasmid self-replicates, typically unidirectionally, and it replicates through the connection port into the other bacterial cell, and then eventually finishes replicating. And eventually, once the replication is finished, the cells break apart, and you have the F positive cell has a copy of the plasmid, and then the formerly F negative cell now has a copy of the plasmid itself. And remember, now that it has a copy of the plasmid, it is now competent and it can actually perform conjugation with another bacteria. Now, just as a remember, most plasmids are self-replicating. They don't. I'm sorry. All plasmids are self-replicating, and most of them contain um, the genes needed to perform conjugation. Now, some plasmids do lack the proteins to build that mating bridge, but they can transfer if one is present, and this is what we call the mobilizable plasmid. This plasmid can be transferred. This one can't, but by transferring this one over, it causes this one um, um, uh, to become mobilized so that it can then be transferred through the bridge, thus causing a double swap here. So just as a note, not all plasmids can naturally move, but plasmids can induce mobilization in one another. Now, what are some of the effects of horizontal gene transfer? So those are the three processes. It's a pretty big deal in microbes. It spreads very useful genes, and these include antibiotic resistant genes, genes to degrade specialized or unique substances such as oil or rare nutrients, genes to encode resistance to toxic metals, genes that allow symbiosis, and as well as what we call pathogenicity islands, which actually encode for genes that act to allow a bacteria to act as a pathogen. Um, Pathogen islands are actually cool. These are large sets of genes grouped together and they're transferred as a unit. And again, they do confer the capacity to invade a eukaryotic host. And so if you guys were alive in the 90s, there was a E. coli strain called E. coli 0157H7. It was also the Jack in the Box bug, very, very deadly form of E. coli that got its pathogenicity from a pathogenicity island it horizontally acquired from another bacteria. Now, let's talk about homologous recombination because we have to talk about this because all types of our bacteria, um, the, their capacity to uptake DNA from the environment or for a virus requires homologous recombination. Now, homologous recombination essentially is the entering DNA replaces chromosomal DNA. So we have an example here. We have a piece of DNA. It combines homologously, so it's based upon very similar DNA sequences or homology. It enters, again, via transformation, conjugation, or transduction, and it essentially embeds itself into actually movement of DNA within a cell, and these are what we call mobile genetic elements. In particular, we're thinking about actually what's called transposable elements or transposon. And now, there are DNA elements within your genome, as well as bacteria's genome and corn genome, whatever animal you can think of, there's transposable elements. And these that have the capacity to jump from one place in a genome to another. So historically, they're actually called jumping genes, just as a note. And there are a couple different types. We have, in terms of mobile genetic elements, we have insertion sequences, and these are the simplest type of transposable element, and they consist of transposase genes in what we call inverted repeats at both ends that are essentially targets for an enzyme called transposase. And unlike homologous recombination, the integration of insertion sequences into any part of a genome is actually random. So that these insertion sequences can jump in anywhere. Doesn't matter. They just jump in where they feel like. Next up is transposons, and these are actually composed of a gene that is flanked by two insertion sequences. And some transposons are actually what we call conjugative transposons that are actually able to transfer from one cell to another via conjugation. So sometimes in addition to a plasmid, bacteria will swap transposons as well during conjugation. And then finally we have is what we call an integron. It's a very large complex insertion sequence or transposon that has what we call an integrase gene that actually compiles a suite of genes. Typically, it's an antibiotic resistance genes, but it can be a number of different things. And these genes actually get regulated by the same promoter. Now, let's talk about insertion sequences and transposon. So as I mentioned, insertion sequences, so in the very first part here, have outward facing promoters, which are labeled as P here. Well, this one is a Q technically, but it's supposed to be a P. Um, and the idea being is that you can excise or transpose one of these insertion sequences and then integrate it into a new part of the genome, as you sort of see in this basic schematic here. Now, transposons are comprised of two insertion sequences with genes in the middle. So we have insertion sequence 1, insertion sequence 2, and then we have some sort of other cassette of genes, typically antibiotic resistance genes, but it can be any number of things. And you can see we have this transposon, it's excised, and then it's integrated with another part of the bacteria's chromosome. So just jumping from one part of the chromosome to another. And so just to sort of show you an example of the effect of transposons, we're going to talk about a bacteria called Klebsiella. It's one of the causes of bacterial pneumonia. Pneumonia is just a pretty broad classification of diseases. It's basically anything that causes like a severe respiratory infection. And Klebsiella is just one of the bacteria that does it. Now, there is a gene called AMP-C, and this gene AMP-C is responsible for resistance to ampicillin, which is a beta-lactamase. It's just a class of antibiotic-resistant genes that targets the psyllin classes, so amoxicillin, ampicillin, penicillin, things like that. Now, AMP-C is an interesting case here. So we have AMP-C on the chromosome. It's poorly expressed, and there is essentially no resistance phenotype. Now, the bacteria can have mutations that make the promoter of this gene, so what tells the gene to be transcribed, stronger. Well, when it's well expressed like this, it confers resistance. And once it's well expressed, a transposon can actually pick up AMP-C, and that can cause it to enter a plasmid. And once this enters a plasmid, it can then be transferred to other bacteria. To other bacteria via conjugation or picked up directly from the environment. So again, similar structure, but you can essentially change a gene by just modifying slightly its genetic structure. It's kind of a weird thing, but it's a really common thing observed in our microbes. Now, let's talk about integrons and multi-drug resistance. So we have what are called gene cassettes. So we have X and Y, and we have an integron here. And we have an enzyme called integrase. And so when we have a couple of genes, so we have a couple of genes for antibiotic resistance, all these genes are under what we call a common promoter. And we'll talk about this in a few classes about what a promoter is, but just to know it tells the bacteria to transcribe a bunch of genes together. Now, the idea being is you can have genes together on one of these integrons, and because they're under the same promoter, and if they're flanked by the right regions, can actually cause the enzyme, which we call integrase, to essentially pick them up together and maintain them together. So when we're thinking about multi-drug resistance penicillin gene here, which is AMP. So now you might be asking yourself, what is the frequency of horizontal gene transfer? Well, under ideal um, under ideal conditions in the laboratory, it's about 1 in 10,000 cases. So not super common, but not super rare. Now that's the laboratory. What's the frequency in nature? Well, it's kind of hard to estimate. So the first one, we can actually look for identical genes in non-related species, or we can examine what we call the G and C content. Now, if you remember back to your first science class for how DNA works, um, adenine binds with thymine in your DNA, and guanine binds with cytosine in your DNA. And by looking at the percentage of your genome that is G plus C, you can look at where things are required, because the sort of the function of G and C content is within, if you have genes all from one bacteria, the percentage of the DNA that is G plus C is pretty consistent. And so if you're looking across a bacterial's genome and you find a consistent amount of GC content, but you find one random gene that has a very, very different GC content, well, you can assume, because it's very different from a bacteria's natural GC content, that it's likely acquired from another microbe. And so let's talk about an example here. So we're looking at an example of Bacteroides. And so it's a major genre within the human colon. So it's about 25% of the bacteria found in your colon. And so this is a kind of an interesting little study. They looked at isolates pre and post antibiotic use and looked for antibiotic resistance genes. So they looked at the source of the isolates in the community pre-1970, in the clinical setting pre-1970, the community from 1997 to 1996 to 1997, and then the clinical setting from 1980 to 1999. And we have 4 antibiotic resistance genes here. So we're seeing the number of isolates is essentially, one level here, so 69 and 23 total. And then later, again, about 20 years later, we're up to 102 and 87. And it's a huge jump in the clinical setting. And it does vary. So there's lots of TET-Q genes, which is tetracycline resistance, and much less, those are pretty common early on and then later in the example. And then you can look at these other genes. So a couple other different types of antibiotics, and you can see they're almost absent in the population early on. And then later on, we see there's a pretty large explosion. Now, it's kind of cool is that you can actually look at the genes from this study, and they actually were confirmed to be carried on a conjugative transposon. So pretty cool. Pretty cool sort of example of how we can see this in actually a clinical setting in real time. Now, let's talk about interdomain, so movement of genes from bacteria to plants or bacteria to humans. And the case we're going to talk about is actually crown gall disease, which is caused by a bacteria called Agrobacterium, one of the most important bacteria in molecular biology, just as a note. Now, gall is just a bacterial infection that we see. Anytime you see like tumor-like structures on a plant, it's what we call a gall. Now, the way our bacteria works, what it does is it essentially picks up sources from the environment that stimulates the expression of what we call a mating bridge between the bacteria in the host. And these compounds from the environment are actually secreted by the plant. Now, what ends up happening is it causes the bacteria to stimulate, again, form this mating bridge, and then essentially transfer what we call its tDNA to the plant. And this directs the plant to actually multiply and produce tumors, which then produce what are called opines, which then the bacteria then eats. So it's what it's doing, it's infecting the host, reprogramming the host, again, with horizontal gene transfer to make a tasty snack, which looks like this, for the bacteria to feed on. So a pretty natural process that happens in nature, but it's actually a pretty common thing used to genetically engineer plants. So if you ever had a genetically engineered plant, which my guess is probably everyone in the class has, what ends up happening is that scientists have essentially taken Agrobacterium, modified its genetics so that instead of infecting the plant with a gene to make tasty food for the bacteria, they infect the plant with a potentially helpful gene, so maybe it will make the plant resistant to in particular, a pesticide or a particular pest. So we can use this horizontal gene transfer between domains to essentially do important things for agriculture. And there's all sorts of GM plants out there. So cotton with herbicide genes, soybeans with lower saturated fat content, rice with more iron and beta carotene, so golden rice. So there's lots of really powerful examples of how we can use interdomain gene transfer to help humans. And then sort of the final thing about interdomain genome transfer is actually something about the human microbiome. And so as we'll learn later in the semester, microbiomes, or the microbes that are associated with all living that you've lost your capacity to, as a human, to make vitamin K and many of the B vitamins. However, your microbes have this ability, which is why you lost them. And so your microbes actually on your body hold thousands of genes that you have lost because they have them. So you don't need to have them because your microbes do. So it's a fun sort of way of your microbes are pushing on you to lose things, and your microbes are also losing things as well. So it's this sort of crosstalk of, you lose stuff, your microbes gain stuff, and vice versa. And we can actually think of another example. So if you guys remember back from class 1, we talked about the pea aphid and its symbiont Buchnera aphidicola. Um, this individual in its little oh shoot, I forget what these are called. In its little bacterial appendages here, you can see them sticking off the back. It actually houses bacteria in there, and it relies on the bacteria to give it amino acids because it's lost the capacity to make amino acids. So, and this is just a schematic of what this looks like. And here's a really great article if you want to sort of dive into it more. And this is just a sort of a final helpful table to sort of give you reminders of all the different things we talked about, from transformation and transduction and conjugation, as well as insertion, sequences, transposons, as well as integrons. So let's just summarize this first part. So prokaryotes can transfer DNA from one mature cell to another via horizontal gene transfer. Horizontal gene transfer happens via 3 mechanisms: transduction, transformation, and conjugation. Horizontal gene transfer can be a powerful way to confer many new benefits for the recipient, including antibiotic resistance genes, new metabolic pathways, pathogenic traits, and many, many, many others. Remember that DNA can also jump around within a genome via regulation. So the questions we're going to try to answer is, how do bacteria regulate their gene expression, and how does that change in response to the environment? What is the role of the sigma factor, and how does it work? And how does the lac operon work, and what roles do activators play, activators and repressors play in its operation? So I'd like to start this part of the lecture with 2 examples. So bacteria, remember, they need to respond fast to the environment. So the first example is a bacteria that's living in a pot of dressing at a salad bar suddenly is transported into a pool of stomach acid. Example 2, a bacteria living inside an insect host is suddenly injected into a human arm. What do these 2 have in common? Well, what you should pick up right away is that both involve immediate changes to different environmental conditions. So the question should ask yourself is, how do bacteria adapt to this brand new environment? So that's going to be our sort of discussion, this idea of adaptation and changes in the activity of bacteria in a new environment. Now, bacteria adapting to the environment differs depending on the timeline. It's seconds to generations. A long-term strategy for adaptation is what we call a mutation, which is just a change in the genetic code of the bacteria. And mutations can be very, very beneficial, but also very risky. So mutations have some issues because they occur randomly, and some new traits require multiple mutations, and it's not a reversible thing. Once you get a mutation, it's really hard to kind of get rid of it. right? If you have a bad mutation, it kind of sticks around. That's sort of the unfortunate thing about mutation. But again, it's a long-term strategy. This is in contrast to a short-term strategy, which is gene regulation. And so we're going to talk about both these today. We're first going to talk about this short-term strategy, and then we're going to talk about the long-term strategy. But gene regulation is rapid and reversible. So controlling gene expression is a very important part of any organism's physiology, not just a bacteria's, but also yours. And so we're going to focus on how bacteria do it. And luckily for us, the way bacteria control gene expression is much, much simpler than the way we control our own gene expression. Now, we have two different types of expression. So we have constitutive expression, where genes are expressed at the same level under all conditions. And then we have regulated expression, which is genes are transcribed only under very specific conditions. And so the question we're going to ask is, how can bacteria manipulate transcription levels? Well, there's one common way. It's by promoting with what we call a sigma factor and activators, or inhibiting with repressors RNA polymerase activity. So we're altering directly transcription. So taking DNA and making it into RNA. And then that RNA is then made into a protein. Now, the main difference between these two types is that the expression is the composition of the promoter. So let's talk about those. Now, the thing about promoters, it is what we call positional information. And what do I mean by this? Well, there's a sequence that marks the beginning of every gene. So the beginning of the gene starts here. So +1 is essentially the first base in the new gene. So we have the C, this part here. So this, what we call the 0 point, and then the gene goes to the right here. Now, anything backwards from this is the negative or downstream of the gene. Now, the promoters themselves are before the gene starts. And in particular, they're what we call highly conserved elements. These are which are ultimately bound by the sigma factor, which we'll get to in a second. And these have what we call -10 and -35 elements. And so we can look at the promoters in E. coli over a bunch of different genes. You know, the ribosomal genes, they're associated with bacteriophages, there are all sorts of different types of genes here. What you'll notice is that the promoters are similar in DNA sequence, but they're always at the exact same spot at the -10 and the -35 positions. That's an important thing, -10 and -35. Now, sigma factors themselves are specialized bacterial proteins that bind to RNA polymerases. Now, remember, RNA polymerase is an enzyme that transcribes RNA from DNA. So this is what starts transcription, what does the process of transcription. So making messenger RNA from DNA. Now, what sigma factors do is they recognize the promoter and guides the RNA polymerase to that target DNA sequences. Remember, they're targeting that -35 and that -10 elements. And I also mentioned that some genes, like the ones that encode ribosomal proteins, actually need to be turned on all the time. So thus they share what we call a common sigma factor. So let's talk about this. So we have an RNA polymerase in what we call the this transcriptional complex. It's a complex of multiple subunits, multiple enzymes. These promoters are saying, hey, come here, very specific sigma factor, which then recruits the RNA polymerase. That's the idea. And we could look at it, what this looks in this orange glob here and this like tealish blob here. So that's what it looks like in sort of a 3D structure. Now, different sigma factors do recognize different promoters. Sigma factor abundance directly affects RNA polymerase activity. So making more of a sigma factor directly leads to more transcription of that associated gene or genes. And typically environmental sensing systems control sigma factor production. And so there's multiple different types of sigma factors. So we have a few of them. So RPOD, H, F, S, and N. And so this RPOD actually recognizes most genes. RPOH recognizes heat shock genes, whereas RPON actually only recognizes genes for nitrogen metabolism. But you notice that they all attach to the minus 35 or the minus 10. But I will say this RPON is special and it targets minus 24 and minus 12. It's a special case here. But the important thing to note here is that if you have a sigma factor that can actually turn on multiple genes, it allows the bacteria to encode to coordinate function. So for instance, if you're a bacteria and you get dropped into a hot environment, well, having one sigma factor that controls all your genes associated with heat shock, you can turn them all simultaneously and respond to a heat shock much faster. Now, sigma factors themselves can be used to regulate gene expression in response to an environmental stimulus. Now, let's talk about an example in E. coli. Now, sigma factor H is needed to turn on heat shock genes in E. coli. Now, at a low temperature, so 30 degrees for E. coli, sigma H is poorly translated due to mRNA structure. So under very low temperatures, our sigma factor H, which is transcribed from the RPOH gene, it has these kinks in it. And when it has these kinks in it, it's poorly transcribed, and it makes poor proteins, and it's eventually degraded. However, at higher temperatures, the greater production due to more translation. So what ends up happening is at higher temperature, these kinks essentially come apart, leads to a straight mRNA, which leads to the production of proper sigma H proteins, which leads to the bounding of that sigma H to heat shock genes, recruiting RNA polymerase, and then transcribing all the genes associated with heat shock genes. Pretty fun, right? In the absence of heat, it doesn't work very well. In the presence of heat, it works fantastic. Now, you don't have to rely directly on the environment to control regulation of your transcription. You can use regulatory proteins, which are called repressors and activators. And these proteins actually physically interact with the DNA itself or with RNA polymerase to alter patterns of transcription. Now, a promoter is a region of DNA upstream of a gene that initiates the transcription, and it's the site where RNA polymerase binds. A repressor is a protein that inhibits the expression of genes, often by blocking the attachment of RNA polymerase to the promoter, thus preventing transcription. An activator is a protein that increases the expression of a gene, often by helping RNA polymerase bind. And then finally, we have an inducer. It's a molecule that binds to a repressor or an activator and acts to regulate gene expression. It will essentially turn on or turn off a repressor or an activator. Oh, sorry, one more. And an operator. It's a region where a repressor can bind and block RNA polymerase. So that's our terminology. Let's talk about this. So first, we have a repressor. So we have a couple scenarios to talk about. So in scenario one, the repressor will bind to the DNA and represses the target gene. And this inducer causes repressor to release. So we have a repressor. It binds to a gene, preventing the transcription. But if you have a ligand, so it's what we call an inducer, it can then bind to the repressor, causing the repressor to come off and allowing the gene to be transcribed. Now, scenario two for repressor, we have a repressor core processor complex activator itself binds to the ligand, which is an inducer, and this complex binds to the regulatory sequence and activates the target gene, thus transcription happens. If you remove that ligand, you stop transcription. So that's the sort of the backdrop we're working with here. Now, bacteria like to be efficient, so we have what we call gene clustering. So if you have genes that encode proteins that are associated with the same exact process, so we've been using heat shock, that's a great way of thinking about it. They're actually found clustered together in the genome. And this is pretty common. We see this with antibiotic resistance and pathogenicity genes. But these genes, when they're clustered together, can be controlled by the same regulatory machinery. And this makes them much more easier to be turned on and turned off as a group rather than being regulated individually, which makes them much more efficient. And when you have a bunch of genes that are involved in a similar process together on the same genome, regulated by the same machinery, we term this what we call an operon. And the classic example that everyone uses when they talk about operons, and we're going to talk about here, is the lac operon. And it's very, very, very well studied. The original researchers won a Nobel Prize for it in 1965 and is still an active area of resource. But the lac operon is required for the metabolism of lactose in E. coli. And lactose is a sugar. It's the primary sugar in milk. If you're lactose intolerant, you can't break it down, so your lac operon doesn't quite work. So just as a fun fact. Now, the lac operon regulates a group of 3 genes: lac-Z, lac-Y, and lac-A. So we have Z, Y, A. And these encode for enzymes that transport and break down lactose into smaller components, which include glucose and galactose. And then this also has a operator here, a promoter, and what we call a cap binding site. So let's talk about what happens in the absence of lactose. So in the absence of lactose, the lac repressor is bound to the operator, which prevents RNA polymerase from binding to the promoter, which leads to no transcription. And you should ask yourself, why is this good? Well, it's good because, well, if this is bound, that means you won't produce these genes, right? That's good. Now, when lactose is present, the lactose is available, an inducer, what we call allolactose, which is an isomer or a similar, which is a similar compound as lactose, binds to the lac repressor, and it causes it to essentially fall off from the operator. And then once that happens, the RNA polymerase itself can bind to the promoter and then begin transcription of lac-Z, lac-Y, and lac-A. But wait, there's even more. There's way more. Now, chemistry is a two-way road. If you're breaking down lactose into glucose and galactose, chemistry will tell, if you've ever taken chemistry course, it will tell you that the end product of a chemical reaction can oftentimes control whether a chemical reaction can occur. So we should ask the question now, what happens if there is glucose present? Well, glucose is a better energy source than lactose. So if glucose is present, the cell will actually want to use glucose over lactose first. So let's talk about what this happens. So if glucose is absent and lactose is present, the amount of what we call cyclic AMP or CAMP is inversely proportional to the amount of glucose. So when CAMP is very, very high, glucose is very, very low. When CAMP is very, very low, glucose is very, very high. But CAMP is an inducer. What it does is it attaches to the catabolate activator protein, or CAP site, which can then bind to the CAP binding site. I know it's getting kind of complex, but the diagram does a lot of justice down here. Now, what CAP does, it acts as an activator, and it helps the RNA polymerase bind, which leads to way more transcription. So the idea being is when CAMP is high, glucose is low, and when glucose is low, it binds to the CAP site, or actually glucose is absent, I should say, not just low. CAMP is high, it binds to the CAP, which then allows RNA polymerase to transcribe more efficiently and more actively, which means that if there's no glucose, it tells E. coli, use lactose. So that's a good thing. Now, again, as I mentioned, low glucose, high CAMP, high glucose, low CAMP. Now, you transcription of the lac genes to be significantly lower. So here's a nice little handy table for how lac works. So the lac operon is controlled by lactose and glucose. So the transcription levels do vary according to their availabilities. So when we have lactose present and glucose is absent, there's lots of transcription, right? Because glucose is absent, that means CAP is binded, which means transcription goes really fast. When lactose is present and glucose is present, again, it means we have very low CAMP, means CAP is not bounded, and transcription is heavily suppressed. So there's some transcription, but not a lot. Now, when lactose is absent, even if glucose is present, there is no transcription at all. So this, again, this is a system that is regulated not just by lactose, but also by glucose. And so does it actually work? What would the growth curve look like if you were using the system? So let's look at this. So we have the log bacterial cell culture and the time in hours. What would this look like? What would this growth curve look like if a bacteria was switching from glucose to lactose consumption? So it actually works really, really well. So this is from a 2014 paper. So cellulobiose is actually a very similar sugar to lactose, and its genes are regulated almost identically as they are to lactose. And what we see is this is the bacterial growth, so the density of bacteria, and this is the time in hours. So what we're looking at here is essentially the amount of glucose present. Um, so we're looking at growth um, on so red line is just glucose, blue line is cellulobiose, so that's similar to lactose, and the green line is cellulobiose and glucose together. Now what you can see here is regular growth curve for cellulobiose, regular growth curve for glucose, but when we have both present, you'll notice that we have growth of the green curve. Then there's this sort of pause where we have what they call a dioxic lag phase, and then it starts up again. So you can physically see transcription is happy for glucose, it stops for a while, then switches directly to working on cellulobiose. Pretty cool, right? You can see the growth curves almost mimic perfectly here early on. Then there's the lag, and then the green one catches eventually up to the blue curve. So it's pretty cool. It works in real time. It's kind of a fun little thing to see in real time. So let's summarize this part. So overall gene expression can be altered by regulating transcription. Sigma factors are key regulatory elements that tell RNA polymerase where to bind, and different sigma factors can be made in response to the environment, such as the heat shock protein, sigma factor H, as we discussed. Now, transcription as a process can be altered by direct interactions with proteins, including our activators and repressors. And these elements are turned on with what we call inducers. And then finally, operons, including the lac operon that we discuss, are great ways to cluster and regulate many genes at the same time, which is more efficient and allows the bacteria to respond faster to the environment. So that's short-term adaptation. Let's talk about long-term adaptation. And how do bacteria acquire new genes? We'll have just a brief refresher because we already covered this in this lecture, but we'll give you a refresher. Then we'll talk about how genomes evolved, and then we'll talk about a case study with E. coli. And if you want to learn about this case study, this great website will tell you all about it. Now, remember there are three types of horizontal gene transfer: transformation, transduction, and conjugation, right? Transformation is direct uptake from the environment. Transduction is virus-mediated ones, and conjugation is direct cell-to-cell transfer of the DNA. Now, bacterial genomes evolve via several different mechanisms. So the first is gene duplication. The second one is whole genome duplication, which is particularly rare in bacteria, but there are many bacteria, such as Cynorhizobium meliodi, that actually do this. Transposable elements, mutation, and finally genome reduction. So let's talk about a couple of these examples. But first, we're actually going to start with natural selection, and we're going to start with Charles Darwin. And so Darwinian evolution dates back to the 1850s, and Darwin observed a couple key things. So first is that individuals in a population have traits that vary. These traits are inheritable. When an organism reproduces, it produces more offspring that can possibly survive in the environment. Competition is inevitable, and species generally suit their environment. So this is what Charles Darwin observed back in the 1850s, and it formed the basis of his theory of evolution. From all these ideas, he proposed three key things. This idea of descent with modification from common ancestors. Second, we had natural selection was the mechanism, i.e. the environment selects for traits. And the final thing he proposed, evolution was slow, occurring over millions of years. So this is what Darwin proposed, these three things. And the important one here is descent with modification. You have an ancestor that passes traits on to the next generation, and those traits can be modified. And how are they modified? Well, they're modified by the ways we're going to talk about, and they're ultimately selected for by the environment. So if you have a trait that's passed down and it's beneficial for the environment, natural selection selects for that trait and allows an organism to reproduce more, thus passing that trait on to the next generation. Now, the way Darwin talked about this was in the Galapagos finches, and he looked at beak size. And what he found is that beak size correlated to what types of food these finches would eat. Now, the idea being is that there was some ancestral finch that landed on the Galapagos Islands. It encountered all these different food sources, whether it was plant material, insects, cacti, or large insects, small insects, you name it, tons of different food sources. Because there was variation in the original population that came, the environment, so the different food source, selected for different size beaks, and selection for the different size beaks led to the evolution of these different species. And you can see just physically that these species look very, very different. And they look very different because of their coloration as well as their beak size. And that beak size, again, was selected for by the environment. Um, and so that's sort of the basis for Darwin's work. The environment selects. So let's talk about how these things occur at the genetic level. And so let's start off uh, mutation. So mutation is, remember, just a single mutation is just a change in the DNA sequence. And remember, which is a fantastic antibiotic to kill um, um, a fantastic antibiotic to kill and treat tuberculosis. But it was interesting is that early on in the clinical setting, tuberculosis actually had a single base pair mutation in a gene, RPOB, which is RNA polymerase, which led to resistance. So what exactly happens here? We had a normal population of rifampin susceptible tuberculosis, and we had a couple bacteria that had this mutation in their RPO gene. Well, when this population gets exposed to rifampin, what ends up happening is it causes the death of most of the susceptible tuberculosis, leading to the proliferation of mainly resistant bacteria. So again, a small subtle change in the genome led to a complete change in the population. Because the important thing to know about evolution is it doesn't act on individuals, it acts on a population. So next up here is actually going to be genome duplication. And so a great example of this is actually the yeast Saccharomyces cerevisiae. It's a very, very important industrial fungi. It makes bread, wine, beer, all the good things in life. Mostly bread for me. I'm a big bread guy. Um, and what ends up happening is we have two modern Saccharomyces relatives, Saccharomyces castillii and Saccharomyces cerevisiae, and they had some sort of ancestor. And this ancestor had about 5,200 genes. What ends up happening, it had a whole duplication of its genome event, leading to about 10,000 genes. Through time, it lost thousands of genes until it got down to about 6,000 genes. And then each fungus lost different genes. So this one lost about 150 genes. This one lost about 230 genes, which led to the speciation and the very distinct differences between these two fungi. So that's genome duplication. And the final one we should discuss is gene duplication. And this is something that's super duper common in bacteria. Um, and it's also pretty common in humans. So if you ever wondered how you got your different types of hemoglobin, you got it this way. Now, the idea of the way gene duplication works is you have a gene, it gets duplicated in your genome on accident. Now, what ends up happening, you either have slow sequence diversion or slow mutation, which leads to the maintenance of the original function of the gene. So you just have two copies of the exact same gene, and the mutation rate's low, so nothing happens to the extra copy. Or we have very rapid sequence diversion, which leads to either a new function, like a related function, but slightly different. So in the case here, we have the human globin genes as a great example. Or it leads to a complete loss of function, or it leads to a brand new function. So the human globin genes are a great way to show related function, but a complete new function we can see with antibiotic resistance genes all the time. So sometimes you'll have a gene mutate enough after it's been duplicated to give you resistance to a new antibiotic. So these are the ways that our gene coli and for understanding biology, but it's also a pathogen. So for those of you that have ever, if you were paying attention, what 2 or 3 years ago, a bunch of people got infected by E. coli in their Chipotle. So it's both a good thing and can be a bad thing. Now, Richard Linsky, he's a scientist at Michigan State University. He started an experiment with E. coli in 1988, so 32 years ago. And what he did is he grew 12 identical lines of E. coli in broth just to see what happens. If you have a few minutes, please take a moment to watch this YouTube video. It's a pretty good YouTube video about the topic, just as a note. Now, what did Lenski do? Well, this is a little bit outdated picture, but I can't find a better updated one. But his experiment has examined 58,000 generations of bacteria. This is as of 2013. He's actually up over almost to 80,000 generations of bacteria, which leads to him seeing about 6.6 generations of bacteria per day. Now, what do I mean by generation? So a human generation is I grow up, I marry someone, and we have a kid. That's a generation, right? That's something that takes decades for humans. We're seeing 6.6 generations per day for his E. coli. Because remember, bacteria reproduce very, very quickly. His experiment has seen over 10 to the 14, or about 100 trillion bacterial cells. He has 12 replicate population. He has a freezer full of well over 4,000 vials, which is just crazy. He's used over 10,000 liters of media, and all these things have been housed in well over 6 freezers. Remember, this is a long-term experiment. It's cost well over $4 million, which is a lot of money, I know, but we've learned a lot of really important things that we'll discuss very shortly. And it's involved about 75 person years of work, which led to the training of 30 graduate students, hundreds of undergraduate students, tons and tons of papers, tons and tons of scientific applications. So it's a huge experiment. And the how important it is going to be lost in this class because, this to explain all the things here, it would take days and days and days of lectures to go over them. But a lot of information he has generated. And I don't want this to get lost about how important this experiment is. But we're going to talk about a few things in terms of how it's related to bacterial evolution. So let's dive into the experiment. What the heck did Lenski do? So he grows his 12 E. coli in a broth with glucose and citrate, plus all the other nutrients that E. coli needs. Now, what he does is after this E. coli grows for 24 hours, he takes 1% of the volume and transfers it to brand new, fresh media. So it has all the nutrients E. coli needs. And he does this every 24 hours, which again leads to about 6.6 generations of E. coli every 24 hours. Now, and then you can look at the timeline of this if you want. Timeline, thousands of generations. And what he does is he actually, the cool thing about E. coli is, in all bacteria, is that you can freeze them um, and resuscitate them once you freeze them. So that's kind of a cool thing. And what this does is it creates actually a fossil record. So he can look back in time to E. coli fossils, but he can bring those fossils back to life. So picture it like, you're studying dinosaurs and you can take your dinosaur fossils and bring them back to life a la Jurassic Park. That's what Richard Linsky can do, which is really cool. But the cool thing is that E. coli can use glucose as a sugar, as an energy source, but they actually can't use citrate aerobically, so in the presence of oxygen, naturally speaking. They can use citrate anaerobically when there's no oxygen, but there's always oxygen in his experiment, which is pretty cool. So that's an important thing. It has citrate and glucose. E. coli can use glucose, but cannot use citrate in the presence of oxygen. So again, this is the concept, the framework. E. coli, it grows for 24 hours. He transfers it, he freezes some. And the idea being is if some mutation happens, you can observe that mutation happening real time, but you can also have a fossil record of it. And you can see mutations popping up over time as well. And you can catch those in real time, and you can capture them in the fossil record. So it's a really cool system. It's a really cool framework. I like it a lot, if you couldn't tell by now. So let's talk about what we know. So at 30,000 generations, well, actually, let me take this back. We have three distinct clades of E. coli evolved. So they're color coordinated here. And the important thing about this is this red clade here. And this red clade is what we call the CIT plus clade. And it evolved around about 30,000 generations. Now, CIT plus, which what is CIT plus? Well, CIT plus um, is a E. coli that has a mutation that now allows it to use citrate as a carbon source in the presence of oxygen. Now, what do you mean by that? Well, remember, before, at the start of this experiment, so all the way back at generation 0, it could not use citrate in the presence of oxygen. Fast forward 30,000 generations later, a mutant in one population evolves the capacity to use citrate. So it basically took a trait it didn't have and evolved it. And evolved it inside of a glass jar, which is wild if you think about it, right? You're taking it's like me, not having the capacity to eat battery acid, and then I just sit in my room for 30,000 generations, and my family now has the capacity to eat battery acid. That's what we're talking about here. Really wild stuff. So how did it happen? Well, first the duplication had to occur. So the normal CIT operon looks like this. The CIT plus operon looks like this. So it has this is what it normally looks like: CIT G, CIT T, and then MA and R and K. What ends up happening, you have a exact duplication of um, the CIT operon. So you can see it's an exact duplication. Once that duplication happened, mutations had to mutate the second operon. Now, this original operon was designed for the processing of citrate in the absence of oxygen, just as a note. So you're taking something that used citrate in the absence of oxygen, you let it mutate or just change over time, that then let them acquire the capacity to use citrate in the presence of oxygen. And then finally, over time, that function was refined. So originally it was kind of sloppy. I mean, it wasn't really good at using citrate, but as evolutionary time happened, as natural selection took over, as more mutations occurred, the process got much more refined. And so citrate utilization started off relatively primitive, but over evolutionary time, it became more advanced, it became more efficient. And now I really like this quote. And so it's really thing. So it says, before a new function can arise, it may be essential for a lineage to evolve a potentiating genetic background that allows the actualization of the mutation to occur or the new function to be expressed. Finally, a novel function often emerge in rudimentary forms that must be refined to exploit ecological opportunities. This three-step process in which potentiation makes a trait possible, actualization makes the trait manifest, and refinement makes it effective is probably typical of many new functions. So what this is saying is the potential has to be there. So the potential in our case was using citrate in the absence of oxygen. The actualization was the mutation happening that allowed the citrate utilization in the presence of oxygen to occur. And the refinement is what made it better, made it more efficient over time, made it better functioning. So it's a really, really fascinating thing. So what else did we find over many thousands of generations? Well, relative to the ancestors, E. coli actually became more fit over time. So you can calculate relative fitness. And as you see, this curve increases over time. And what does fitness mean for bacteria? Well, fitness just means it can grow faster. That's all. So it was getting much more fit over time. So it's becoming better adapted to its environment. Makes sense. The other thing, it got fat. So you can see that the cell volume over the number of generations is that the cell volume increased over time. So the E. coli was getting fatter. And just as a note, the units here is femtoliters, which is 10 to the minus 15 liters. It's very, very small cellular volume. What we were also seeing is that the colonies themselves on the plates were getting much larger. And then the other thing we're also seeing is that the E. coli genome had numerous other mutations, not just related to cell size or the citrate utilization. Mutations were happening all over the genome. And there's actually some new data that's coming out that's suggesting that many of the functions are heading towards a loss of function. And so you should ask yourself why. Why, if in a static environment, why is E. coli mutating and why is it losing function potentially? Well, the idea here is that If you're a bacteria, you're trying to streamline your genome. You're trying to make yourself grow faster, produce offspring faster. And if you don't use a function with bacteria, over evolutionary time, you will eventually lose it. And so this is true not just for bacteria, this is true for humans. We lost the capacity to produce vitamin K as a species, as we talked about earlier in this class. Well, the reason for was that the reason for that was because we weren't using it, because our bacteria in our gut were providing vitamin K for us. So as we didn't use that function over time, we lost it. So that's what we've seen with E. coli too. As it stops using functions in this experimental environment, it's actually starting to deteriorate and starting to lose those functions. Anyways, there's lots of cool stuff that this experiment can learn. If you want to learn more, again, please check the website I linked at the very beginning and I recommend diving into some of this literature. It's absolutely fascinating what you can learn from just studying E. coli um, just in a test tube. It's really, really cool. So just to summarize this, there are numerous mechanisms that drive the evolution of bacterial genomes, but ultimately, bacteria, like all other organisms, are just responding to their environment. And our bacteria, as we see, just can do it faster. So with that, if you guys have any questions, please do not hesitate to reach out. I do hope that you learned something from this sort of longer lecture than we're used to. And if anything in this lecture piqued your interest and you had any questions about it, please reach out to me. I'm more than happy to discuss them with you. But with that, I hope you guys have a great week. Take care.